Hello, everyone, and welcome to today's live broadcast, a keynote presentation, NCATS Automation Capabilities, Where We Are and Where We Are Going. It is presented by Sam Michael, the Director of Automation and Compound Management at the National Center for Advancing Translational Sciences, NCATS, at the National Institutes of Health. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I'd like to remind everyone this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please click on the Help Desk button located in the promotional board at the bottom center of your screen. Or use the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Sam Michael. I will now turn the presentation over to him.
Judy and LabRoots, thank you for the opportunity to give this presentation, which is NCATS Automation Capabilities, Where We Are and Where We Are Going. I wanted to start with the history of how NCATS was formed. We originally started in 2004 as part of the NIH Roadmap for Molecular Libraries Initiative, which was dubbed the MLPCN. And part of this was to actually develop new chemical probes for basic research and leads for therapeutic development, particularly for rare and neglected diseases. And even though we started that in 2004, we really continue that tradition to this day. Also, part of our mission was to make sure that we had new paradigms and applications of HTS for chemical biology and chemical genomics. We were part of NHGRI up until January of 2012, actually with the Affordable Care Act. Passing the law was officially NCATS' birthday, so December 23rd, 2011 is when we became NCATS, and we're now a center within NIH. So part of our focus has always been with steps in the drug development process, and where we reside at NCATS is really primarily with the translational piece of our institute, not so much the target identity, which is the basic research side, but more from the assay development up to preclinical development. This is more on the intramural side. The extramural part goes further on to the actual clinical, and that's the CTSA program, Clinical Translation Science Award. The focus of this talk is going to be more along the lines of the intramural program, which is really focused on assay development through medicinal chemistry, at which point typically we hand projects over. And so part of this, and part of the reason for the form of a foundation of NCATS is this concept of the drug development valley of death. If you look from that same chart from the lead to clinical, the chance of success goes very far down and the cost goes exponentially up. And it's a little too risky for commercial entities to really invest in this space. And so part of the mission of NCATS was to try, especially with rare diseases, to bridge this gap, to take promising leads, de-risk them by putting some science behind them and validating that there's actually something of substance there, and hand this over to a potential partner and collaborator who can then carry this forward to a clinical IND, and we've had some success in that. So a core portion of this, of what we do in NCATS and really geared towards the automation, is what we call quantitative HTS or qHTS. And so one of the paradigms that was well established at NCATS early on from some of the founders who had done plenty of screening, all over industry and primarily at Merck, is the concept of not screening at a single point. And so what we actually screen at NCATS is we screen at a dose response. And which is important to note is we don't just screen the single concentration, we actually generate a dose response curve for every assay we run. So what's interesting from an automation perspective is this has been well established, and on the slide you can see several classes, what we're considering class 4, which is in fact no activity, to class 1, which is very potent activity, and all of this code and these classifications are available to the public. But what we realize is from an automation perspective, this made it challenging. So for perspective, if our compound library is 200,000 compounds, but I screen at 5 dose responses, that's equivalent to 8 million compound libraries. So we need a high degree of automation, which we invested in early on, going back to 2005, and we continue to invest. And so when you're looking at these systems here, the one on the right is our primary system. We first got established from GNF, the Genomics Institute of Novartis Foundation, going back to 2005. And this has been our workhorse system, still in use to this day. And it's a much more early generation, you'll see in a moment with the video, large robotic arms. And then the system on the left is a smaller version of that we have for a specific program called the Tox21 screening system. You can see the capacity. We can run several thousand plates, hold them at a time. We can run the millions of wells. We really have a high degree of automated capabilities. But I don't want to say these are legacy systems, but this is a little bit of where we were. We're still here, but what are we going to advance to is the focus of this presentation. And then if you want to see the video now for the high throughput screening system in action, you'll be able to see these arms moving plates from station to station. And so what you saw there was the robotic system moving plates from point A to point B. And really, these arms are very industrial. And that was partly the change in the early 2000s, because one of the biggest failure points of high throughput screening in the 90s was actually guaranteed plate delivery. And these is something that when they switched to these industrial arms, that you actually had a high degree of reliability to ensure that if you wanted to move from station to station for plate, it would actually occur. And these arms have been incredibly successful in what we've tasked them to do. Since we started with many of the assays in the high throughput screening system, we've also since really established a program at NCATS for drug repurposing and combination discovery. And we've had so much demand for this that we've recently, a couple years ago now, have incorporated a smaller scale system simply to just do acoustic transfer. We actually have another device we just got, which is a lab site system. We have both EDCs and lab sites for a variety of reasons here. The focus of the EDCs is more small molecule. The focus of the lab sites is more biologics. But this system is an example of what we're looking at here. You can notice one of the primary differences between this and the previous video is this system is open. There's really no need for guarding because they've since changed robotic arms from those large industrial arms and all the safety requirements to these direct drive arms, which are much more user-friendly. And you can see on the image on the right where it says one robotic arm, it's actually on a track. So you get a fair amount of range of motion from that actual arm without the safety considerations. And part of this is also our methodology at NCATS and something to consider of how you can modularize a process. And what I mean by that is you want to actually separate the process from different steps potentially. You don't need to automate everything. So if I just need to produce a large amount of combination plates, and I'll get to that in a second, I don't need that all on the same system. I can have a separate system work in a modular fashion to produce those plates to act as an input. And so what we're doing here, instead of very large compound libraries, is we actually have a small targeted library. This has been published on, and MIPE stands for the Mechanism Interrogation Plate. It's since expanded from one plate to actual couple thousand compounds. And we run these in combination to look for therapeutics really geared towards oncology and very cell-based assays here. And so what this looks like is instead of having a single point or a single dose response curve, we actually run in combination two competing dose response curves for two of these drugs. We've actually done 3 drug combinations. And what we're really looking for here is the synergy. And so this is another example of some of the challenges that we're facing at NCATS is we have to support the legacy projects, which are with our traditional chemical library. And then as we expand into new biological space and new assay needs, our automation has to account for that. And so part of our model has been to modularize what system performs what task, so not all burden gets put on one system, even though it's capable of pulling it off. And so let's give an example to describe how these schedulers work, how these systems work, and how you can actually manipulate them to actually do more complex assays, but also take a step further from automating something and actually with the goal toward eventual autonomy. And so here's a very basic enzyme activity assay. Is you have enzyme and substrate in the presence of a compound. They incubate and they produce a product. To that product, you add a detection reagent. You bring that plate with all those reactions taking place, and you do a luminescence read to measure what that activity is. And this is just from a workflow perspective. And so, as everyone's well aware of in this industry, most of all the reactions take place in a micro well plate. Now, for the MLPCN, given that the MLSMR, the size of the collection by the end of it was several 100,000 compounds, we decided to miniaturize the 1536 well plates, and a lot of our automation is geared towards that. What's very interesting is we get to more complex biological systems with regards to 3D spheroids and even tissue printing type initiatives, is the density of the well plates actually going opposite. We're doing 1536, we're doing a lot more 384, some 96, we're even doing some 24 well plates depending upon the actual complexity of the structure. But the well plate still remains the workhorse of where all of these reactions take place. So if I look at this system and as a series of devices, you can consider this as the overall peripherals that control what methods you're able to run, meaning what steps you can perform in your assay plate and what kind of experiments that you want to have. Everything from reagent addition in dispensers to compound addition, be it a pin tool or an acoustic transfer device, and to a variety of plate readers. Even looking at this overlay here is having a modular system is very helpful. We've since actually have a Ferristar in this system. We have a Hamamatsu FDSS. We've taken the multi-drops off. There's a lot you can do to reconfigure these systems, and I think that's a key when you're considering what sort of automation you should be looking for, but also to have that flexibility that lets you think about experiments that you haven't even thought about when you first purchase these systems. Another important point is not only the system devices, but to consider these overall systems as for what they really are. It's its own independent subnet. And so this is the network diagram showing that effectively one thing we shoot for, not just us at NCATS, but many folks is TCP/IP is ubiquitous. At this point, there's still some serial devices in the random, Ethernet IP and so on and so forth. But by and large, most of these devices have a direct Ethernet connection, so you can envision this as an actual networked device. And that's a really important thing to consider, because if you have a networked device with a subnet of the individual components, but those components can access the primary system controller, you can actually have a higher level of control over your entire system, and your system itself doesn't have to be isolated. It can actually communicate with external applications, or in many cases now with the prevalence of cloud-based computing, actually do automated analysis and spawn new experiments. And we'll get to that in a few minutes. But it's really important to recognize that these systems now are really part of a network. And so if we want to run an experiment that I described prior, and what is our HTS scheduling methodology? Now, we adopted much of this from the original GNF system, which is the one that we showed prior, the tried and true. And so if you have the plates, and if you treat it that the assay plates act as the input to our HTS system, and they have associated control in compound plates as required, because you have to get the compounds into the assay plate. If you consider each assay plate to be an assay object, a method is the set of steps each assay object will run, with each step usually associated with some peripheral device, be it, for example, dispense this reagent at this time. So a group of assay objects running the same method is considered to be an assay, and we say that with a capital A. That's what we consider, that a collection of these objects comprise one assay. With the system we have, you can run multiple assays at the same time. And that's because the scheduling methodology we have is what we call a wait queue and a mutex. And so how does this work? And so at the start of an assay, all assay objects are placed in the first in, first out wait queue. So this is a FIFO, very well established technique. A wait queue is a queue of objects waiting for a lock, which in this case represents control of a peripheral device on the screening system. When the lock is unlocked, the objects acquire the lock in the order of the queue. Getting back to the FIFO, the first one in is the first one to release the lock. So what this will look like is if, for example, that method I gave, and from this diagram here, is you see that there's an unlock there right by that flask, which will represent the dispenser. The first plate, so the bottom plate in this case of the stack, will actually go, and what that'll do is, since it's unlocked, it will go to that device, and then it will actually lock the device so the plate that's waiting for it cannot have access until it's done with that step. Once it actually performs the dispense and moves to the next step, in this case is a read, is the mutex for the dispenser unlocks and the next plate is free to go through. And so objects still in the queue are in a block state until the lock is released. So if you look at this now, the second plate can't go to the reader because the first plate's still in the read step. If we progress that through and the last plate goes to be discarded, it unlocks the reader, at which point the second plate can actually get read and it can complete its process. And so each step or a series of link steps in a method has an associated lock. And this type of synchronization being called a mutex for mutual exclusion, which pretty much means if something has the lock, another device can't access it. And as simple as this scheduling is, it's unbelievably powerful because you can control to the device level and the assay plate level every single event that occurs and when it occurs. And so we'll give some examples of what we've done at NCATS with this, but I really can't stress the point that this is a very powerful scheduling methodology that gives you flexibility with a higher level application if you choose to take advantage of it. And so from this perspective, if you have a traditional HTS workflow, what does it look like? You'll set up your system, you'll load all the plates and reagents, you'll run the assay protocol for each plate. Key point, there's a one-to-one relationship between assay plates to compound plates. Now you remember that we screen at a dose response, meaning we have a set of intraplate compound plates that are there. So say if I have 4 assay plates, each one of those will be 4 compound plates where the compounds are the same, but the concentration is what varies. And their traditional workflow is you just run each series at a time at the start of the assay, and then eventually it'll run to completion. And then if you look at this again from a workflow perspective, this is the overlay of our system. This is showing what one Gantt chart representation would look like for the variety of the actual steps that would occur. So you actually have the enzyme being dispensed first at one of the dispensers. We have the compound at the pin tool. The substrate is dispensed subsequently. They incubate an incubator, and that produces the product, so on and so forth. And this is what it looks like represented with one specific plate, with the assay plate and the compound plate acting as inputs. Now, if you look at this as a Gantt chart, this is what 20 plates look like. And what you can see from this Gantt chart for 20 plates is these plates ran really nice, and they ran nice and parallel, the parallel lines, and you can see with the rate limiting step being the first enzyme dispense, and nice steady flow of how these 20 plates ran. It was more than 20 plates. This is just one window into this. And all of this is tracked and saved in real time. So we actually have a web-based system where we can look at the assay performance so we can see how the assay is performing as it goes along. So here, there's a couple of points I want to make about what makes this possible, especially with 1536. And something that really gets overlooked, and I think GNF should get tremendous credit for, is one of the key changes they made is what we joke around to as the Kung Fu grip. But when we mentioned that in the past, part of the challenge with the plate failures for high throughput screening was dropping plates and not having this. What GNF actually did was they designed a plate transportation system, meaning they made a mechanical solution with this flange and this dovetail joint between the gripper and the flange of the plate. And that really gives this gripper an absolutely secure lock on this plate for placement. So if you consider the math of what NCATS does here, we're typically over 900,000 moves a year, meaning a specific move of a plate from one location to another, and we have multiple of these arms without failure. And that's really impressive. And so, there's all the software in the world, all of the fancy experiments don't mean anything if you can't have that guaranteed packet delivery, as we call that, from the networking side. This is a really nice piece of mechanism they made here. The next step is you put a lid on it, meaning you have 1536, How do you prevent evaporation? A common technique now used as well is the seal and peel model. Many people have plate seals. Many people have the X-Peel now. Very fantastic device that just works like a champ. So you really have to prevent evaporation, especially with some of the longer cell-based assays where sealing isn't quite an option. There are permeable seals, but they don't work as well. And so some of this is just to give a foundation of if you have a stable platform like this, what can you do with it and how can you expand upon this? So many of these systems, you have the capability to monitor events and then, to see what they're doing, but do you actually have the ability to programmatically control multiple assays running at the same time? And the scheduling software that we have allows for that, since we can actually create custom triggers, and then those trigger events can actually fire some process to happen. So let's give an example of this, and this is just showing the monitoring technique, is we have an entire system running. What it creates is the data from the plate reads, which are saved automatically to informatics, and now we actually save those to a cloud-based environment as well. And then we have a controlling application that can in fact act as the overall listener to spawn new experiments. So we can programmatically launch experiments based upon the results of an experiment running on the system. This is important because as more and more folks get involved in phenotypic screening and some of these more agnostic type approaches, you actually, when you hear in the buzzwords of machine learning and deep learning, what does that really mean? Well, if you have a screening system that's set up now that actually can take external input to create and launch a new experiment, it becomes a pretty powerful tool. But how do we actually get this done? What's the programming and what's the methodology behind this? And so what we've decided and what we programmed at NCATS is we really set up 3 distinct event types. You can do them on a per plate, meaning on one plate we'll have what we call a transition step index, meaning a particular step of a method is actually in a position that we can set an event that when that index occurs or that step of the method executes, it can spawn another process or spawn some event. We can do this based upon a per group, meaning a group of plates. When they're complete, something else happens. Or we can have an external application completely control our system as if it just receives an input, assuming the appropriate inputs are there. And so what are some of the examples here? Well, we're going to focus on 2. We're going to focus on the QC process and also how we initiate a new assay and why. And so this is where we're also talking about how we increase the level of control that we have at NCATS and the rationale why. And so one of the first things that we ran into with the TOX21 robot is they were doing quite a bit of beta-lactamase assays, and the CCF4 dye has a percent DMSO on a valve-based dispenser, the Bioraptor. The challenge with this approach is that's really prone to clogging. And so what you're seeing here is the 4 tips of the clog of beta-lactamase reagent. Now, the reason this was so catastrophic when it happened, if I were to wait to the read step to see that this dispense didn't happen, the read step is after a 2-hour room temp incubate, and if my throughput of plates is about 16 to 20 plates an hour, I may have lost 32 plates before I even capture that this dispense problem has happened. So I had to catch this in real time. And so how do we do this? And so what we ended up setting up is we have a series of sensors, and 4 sensors working in parallel that are retroreflective. And so every time a droplet intersects the beam, if you consider it a through beam type approach, it can actually trigger a response. And then if I have a simple IO module, I can actually capture every single dispense as it happens. And if I monitor that over time, what I end up creating is a digital waveform for each tip. And what I'm expecting is if I expect that in 1 microliter dispense, I should be able to count every single droplet for that dispense as it's occurring. And so how did we validate this? Is if you look at this with these valve-based dispensers, it's what they call a spike and hold circuit. And so the spike voltage is what actually actuates or open the valves, and you only need that momentarily. And then if you look at that, the hold voltage is to keep that actuator open. Once the actuator is held open, the period of time it's open is what dictates, and you calibrate this as what the dispense is. And so if you look at this trace, the yellow trace is the hold circuit, the green is the spike. The bottom cyan color is what the sensor is reading in response. And you can see for a 1 microliter dispense, it's about 8.5 milliseconds of open time. And the sensor's measuring that it saw about 8.5 milliseconds worth of actually liquid dispensed. So we showed that we can monitor this. The 5 microliter, exact same principle. The only difference is you see the valve open time is actually extended from 8 to about 30 microseconds. So what we ended up, or milliseconds, I'm sorry. What we ended up doing is we created a Bioraptor fixture, and what you're seeing in the bottom right is you see that tip array, which the Bioraptor, instead of the traditional 9 millimeter tip-to-tip spacing, did this sort of quad approach, which is a little more difficult. But what you can see is we were able to make a fixture that can do cross sections and actually monitor all 4 tips simultaneously. And so what that allowed us to do is actually monitor in real time and count the number of droplets. And so if you watch these 2 videos back to back, is if you play these side by side, is what you'll see in the software screen on the left is it counting every individual peak. And what you'll see on the right is what that looked like on the actual dispenser itself, if you show those videos.
Yeah, the black plate definitely does look better.
We should also get the multi-drop while we're at it.
Yep.
And so what we ended up doing with this droplet detection, because it was very important for us, and one thing we do at NCATS is we actually like to disseminate what we do once we've demonstrated it, is we actually created an SBIR, and 2 awards were issued for this. And then the real reason is now that the plate transportation problem has been solved, a real limitation is the actual dispense errors really cause a bunch of problems. And then this was also the final missing step of data integrity chain to validate the dispense actually took place, and not necessarily at the plate level only, but the tip level itself. And a big part of this too is conservation of reagent. It's not just the loss of the experiment having to rerun it. A lot of these reagents are really expensive. We ran into a problem though, is the Bioraptor is discontinued. And this is originally from Aurora, and then it sold to Beckman. And then even though our biologists here love the Bioraptors, they're no longer available. So what we decided to do is we wanted to build our own dispenser with the same criteria. And we decided in this case to focus our platform around the Mecha 500 robot as the really dispenser platform. And this gets the going forward, instead of a monolithic system, having a more modular approach. Why did we pick the Mecha 500? So if you look at this picture here on the left, you can actually see someone holding this robot. That's the actual size. It's a tiny little 6-axis arm. And what that gives us is the flexibility to actually do a lot of things that a traditional XY dispenser can't do. It has 5 micron repeatability. There's no external controller and a direct TCP/IP Ethernet connection, and can meet all the criteria for Bioraptor. The other real key point about this is if I have the 6-axis arm grip a plate, and that plate acts as the foundation, is I don't need to actually have a stacker and a dispenser, because the robot arm is the stacker and the dispenser in one. And what does this mean? If you look at this image here, is if you see this, is what you'll see is this is our setup that we have where we have an input stack and an output stack. And then you can see our bank of dispensers there in the back, in the middle of the image there with the angle, and it actually has droplet detection integrated. And what you saw there was the dispenser, the robot acting as the dispenser and the stacker in one. And part of the reason, aside from the footprint, we wanted to focus on that as we consider the cost. And let's give you perspective, is a Bioraptor back in the day would cost $90,000. Even today, you have a multi-drop that costs you about $20,000, depending upon 23, really, they keep on going up in price. If you want to buy a stacker on that, you're talking about an additional $15,000 to $20,000. It really depends upon the stacker. So you're committed to $40,000 for just a basic dispenser, or consider some of the other equivalent ones, not like the Bioraptor. If I can combine those two and the total hardware cost for everything you just saw there between the robot arm and all the valves and whatnot is around $16,000. That's a pretty substantial cost savings that you can actually make and actually have a very reliable device. Let's talk about another event type we can monitor, and the reason why for this is if when we describe the actual process of adding assay to a reagent to an assay plate, transferring the compound, adding a detection reagent, and then actually taking an image of this plate to do a measurement, if you know the difference is instead of trashing the plate from before, what if we could actually clean this assay plate and reuse it? So the first step is we did two experiments. We did wash effectiveness for absorption and for fluorescence. And we did an experiment where we really spiked BSA to 1%. And we wanted to measure if we can clean that out of this plate just using aspirate techniques, everything from a suck aspirator or actually to a centrifuge type technique, and then doing intermediate wash steps with 70% ethanol and water. And if you can see from the actual chart here, from the actual plate, is almost completely removed. And so it showed us that we could actually, fluorescence as well, we can really clean and reuse these plates. And that gives us a thought is, if we don't have to throw these plates away and set up an individual plate for every compound plate we want to run, is it really gives you flexibility to start thinking about these systems as a continuous process and not just a batch mode type approach. And so the first step is, how do I determine the actual assay plate buffer size? And that's the total assay duration divided by the rate of plate addition. And there's more complexity you can add to this, but by and large, because if I run 1,000 plate assay, I don't have 1,000 assay plates on the system at a time. I only have that number of plates which are currently undergoing some step. And so if you determine that buffer size, what you can end up creating is a circular buffer. And so what you can do is, with 2 series equates 1 full cycle, is if I extract from the head of my buffer and then insert plates into the tail, is I can have a continuous process that runs without interruption by using far fewer plates. And so if my step is that I have a buffer size in the example I gave that I only need 11 plates at a time, I can only use 22 plates, and I can run however many compound plates I actually want to get through. And so what this looks like, if you change the workflow, if you remember prior, is now the first set of plates will go and they'll get run against the compound plate. The second set of plates will go and they'll run against their compound plates. But instead of the third set of plates running, is the first set of plates will run again, and then the second set of plates will run again. And this process will continue up until the point that the actual assay is complete. And so what does this look like from a Gantt chart perspective? This is an example of a very complex but slow-moving assay, where this assay ran for 7 straight days with 2 assays in parallel, and all that had to be done was just top off the reagents. And this is a tremendous savings of resources, both from time spent loading up 1,000 plates and then throwing 1,000 plates away, and also saving all of that plastic. If I could run over 1,000 plates with 44 plates, I saved over 900 plates. Another example here is what this looks like with 1 singular assay, more traditional enzymatic. And also from a confirmation perspective, is if we ran over 400,000 compounds in 4 continuous days just by topping off the actual reagents, is what was the confirmation rate? From this assay, we actually had 529 compounds selected, 504 confirmed on the actual follow-up, which is 95% confirmation rate, which is really fantastic. And so part of the reason, and another example people will ask is, will you miss anything with this? And we want to give an example of this LDHA project. In this case, if you look at the hit over here in the yellow in the bottom, it wasn't that high concentration. All of these plates were washed. And so there was 40 plates used, and each plate was used 38 times. And we were still able to whittle out the primary lead, which 2 rounds of med chem in, we actually had a very potent compound, as you can see in the chart in the bottom. So the bottom line is you can actually adopt this approach and not impact your science. And then the question in this case is, why is NCATS involved in this? Is really a huge part is, aside from the reducing the setup time and running this as a long-standing continuous process. And remember that when we talk about eventually making things autonomous, you have to show your system can run continuously. Is we've saved over $500,000 in plastic, and not to mention barrels and barrels and barrels of things handing off into a landfill somewhere or incinerated. And so this also led to an SBIR contract. And part of the reason we wanted this is we're cleaning it, but for cell-based plates, because this was really for biochemical, you really don't have this continuous process because many of the cell-based experiments are endpoint reads. So we use this SBIR, and ion field ended up winning. And their technology, their plasmas, they call it, their tip charger systems, is if you imagine it oxidizes the surface and actually breaks up organic compounds and just releases this as a gas state, so with ventilation. And so part of the reason we want to do this is the estimate you can see in this industry, we use 125 million plates per year. And so if you put those end to end, that's close to 10,000 miles in plates every year that we're using. And if you actually map that out, that's actually from Washington, D.C. to Melbourne, Australia, is how many plates. There's a really fascinating article that came out in The Guardian not too long ago that there's enough plastic that it can actually exist in the fossil record. And you can see from this blurb here, 300 million tons of plastic a year. And typically most plastic is used, once and then thrown away. And in our industry, it's much more than that. So part of the reason as well was this focus on cell-based assays. And this is just showing as well that the plates that come in aren't necessarily all of that clean. There's actually contaminants on the plates as you get them. But can we actually show the effectiveness of cleaning a cell-based plate? So what we're seeing here is cells, and we're doing a cell titer glow read here. The plate on the top was actually run on December 17th of 2015. And what you're seeing with a green highlighted box is that's where the controls were, and that was bortezomib, which is very cytotoxic in columns 2 and 3. Column 1 is no cell. The same plate was rerun on January 14th in 2016, and you can see those cells that went from a total kill. Once it was actually cleaned and cells were reseeded and incubated again, you couldn't tell the difference between the used plate and a cleaned plate. So this is really showing that you can actually get these results without really impacting your science at all, which is first and foremost, is you don't want to introduce another variable. Why were we this focus on all of this cell-based screening? The real issue that we're running into, or not issue, is we've done a real shift from biochemical assays to cell-based assays for a variety of reasons. It's just the assay types we're doing, and that leads to a large amount of cell production, which can run into problems. And this was another area that we decided that we want to automate. And so we started introducing, and we purchased our first compound selectee, where flasks and media go in, and what the output is are either flasks with cells that people can prep, or actually a plate incubator where you can actually plate these cells and have those plates ready to go. So back to this workflow model, if we were to envision this again, is if I have flax and culture media in, and the output a 1536 well plate, how could I get that well plate from the selectee to one of our screening systems? And so conceptually, what we thought is, somewhat facetiously at first, is we said, well, what if we use a drone? You know, we had these two separate systems that were about 15 feet apart, and it would really be straightforward if the cell system can send a message, I have a plate ready for me, and deliver that. That would really be something fantastic. So we took this on and we said, okay, what would this objective for this project be? And it really is autonomously move a plate from one location to another. And in this case would be the selectee letting it know through an event that it's produced a cell plate and it's ready. And then for that to trigger an external event for this drone to go and pick it up, and then to bring that over to our high throughput screening system and land it. And remember, some of this was just conceptual, but we wanted to see the technical challenges of what it would take to really have autonomous, not just from an informatics perspective, but actually from a mechanical screening perspective. Is this conceptually feasible? And so what were the components we would need? We need a drone, we would need the gripper assembly, and we need a charging station, because part of what we decided is this drone would actually have to be recharged in between flights. And maybe it's not one drone, maybe it's a fleet of drones. There's a lot of actual software out there already available for flight mapping. And so the conceptual workflow would be, imagine you have a wall mounting recharging station. It would get the command to pick it up from a location. What we're showing here in this graphic on the right is coming from an old centrifuge. Pick it up from the centrifuge and then put it into an actual hotel of plates here. And we picked this for conceptually a challenge to make this difficult, and then return back once it's dropped off to its base station. And so the first drone we picked the Matrix 100 because it was a supposedly open platform, and also it had the ability to add the Odroid onboard computer, and it had a guidance module you can incorporate. We know we needed more than just the onboard drone computer. We also need some system for indoor navigation, at which point that we actually focused on Vicon, where you can actually set up cameras. And what you're seeing in this top right display is where it shows the software in the visualization of how it's mapping the area. It's a little bit of a inverse logic here. The areas in the orange are really what are showing that it has a lot of positional accuracy. The outer edge in the green is where it just doesn't have as much. But in that center area, if you look at that work envelope, you can actually see it's pretty big. If you compare that work envelope compared to that of a robot with a base station, which is in place, you actually have a lot of flexibility. All of this software does is these 4 cameras, it just streams multiple drone locations as TCP/IP data as a position vector, which makes it really powerful. And if you look at this next video that's showing up, is what this will show is the ability for it to track multiple drones at the same time. Visual representation. So once we got that set up, we also decided in parallel to actually try to do the drone recharging. There's a company called SkySense, which has been actually doing this, and they've commercialized this, so this already exists. What you're looking at is our Matrix 100 on the SkySense platform. On the right there is you'll actually see the two electrodes touching the surface. One important point to make is these platforms aren't constantly charged. It's once the drone with a particular key actually lands and makes contact, that's when the charging begins. What you're looking at the bottom right there is just showing that the current switches from high current to very low current and no current once it recharges the battery. We had this battery completely dead and it took an hour and 20 minutes, but that's assuming that the drone flight time is 20 to 30 minutes. We really don't need that long to drop off a plate. So realistically, you would be in the not too long to recharge this. But there was really some problems with the Matrix 100. It was just too big for indoor flight and it really wasn't safe and it wasn't conducive. The rotor wash was so powerful, it actually just had a lot of variables that even with a good control system, we couldn't maintain. So what we decided instead to do a proof of concept is, how do we have a parrot drone, which is a $200 drone, if not cheaper, take the actual shielding off there and introduce a gripper with this. The challenge we had with introducing a gripper, even with low-level control, is it really adjusts the center of mass. And as this video will show, is when the drone takes off, it should go straight up. And I think you'll see it doesn't go just straight up here. When you saw in that video is when you saw it take off is we couldn't control at the root level of the actual firmware how to accommodate for the center of mass difference, and that's where you saw it actually have that wild swing forward. And then part of it you did notice is over time, though, was able to overcome and compensate, at which point we can move the drone around accurately and then actually land it pretty precisely, even just with the Vicon cameras, which you saw there. You can see the tripod in the back, and we have 4 of these cameras. What we decided to prove this conceptually was, let's just do an electromagnet and pick up a washer to show we can do a pick and place, that it's conceptually feasible with a drone, that we could pull this off, and maybe if we're successful, we could issue an SBIR. And so the system components for this were the controlling software that has the Vicon cameras and the associated data that it streams. And then we have our own custom Ubuntu ROS operating system app that we developed that communicates with the drone and gets positions from the Vicon to feed back to the drone to move to positions that we predefined with the Vicon camera system and the fiducial marks. And so what we're able to show once we did that is we were actually able to do a successful pick and place of a washer, of picking up a washer. So you'll see the drone descend over to one table, and what it's doing there with the electromagnet is picking up a metallic washer, flying to another station, and then dropping that off if you watch this video. Was sort of successful, and successful enough that we figured we could do an SBIR to actually see what the response would be, and it was actually tremendous. I think this was one of the SBIRs that we issued at NCATS that we received the most responses ever. It ended up meeting, or we awarded 3 phase 1 proposals, and we wanted to actually see if someone could meet this challenge with this autonomous pickup and delivery of an actual plate, if you can actually incorporate the low-level design. And so as an example, one of the awardees was actually able to come up with their own custom canted rotor hexcopter for all the stability issues that you saw in some of those videos. Is this provides 6 degrees of freedom control, can translate an XY plane while maintaining level altitude. And really what that means in this case is there's no pitch or yaw when it moves. And the total weight of this drone is about 2.1 kilograms, and it's actually has room to shave maybe 400 to 500 grams from this. And so really what this turned into doing as well is the gripper design and integrating that with the control system so it would account for that center of mass. But if you remember back to our previous slide with the old system, is don't reinvent the wheel, is if we know this dovetail joint is very effective and very in what it does mechanically, is let's go with what's known and create a gripper which can be easily actuated to actually pick up a plate, and we know it's rock solid. And so what you'll watch in this video here is you'll actually see this drone take off with the same Vicon camera systems, and this was done at MIT's lab, working in conjunction with Creaire because they were one of the SBIR awardees based upon some of the NCATS specifications. And it's really been a fun project to see here. And what you'll see is actually pick this plate up, take it back, and then put the plate back in. And so if you want to watch this video. And so what you saw there was the slight adjustment with the control system for the center of gravity is when it picked up the plate, you saw it pitch slightly down. And then when it put the plate home, you saw it sort of raise its arm in victory that I did it. But what it shows is with technology currently available today, is it's feasible that you could use an autonomous vehicle to move plates from point A to point B. And we know that this is actually very preliminary, and there's challenges such as rotor wash. but it introduces the concept of autonomy of systems communicating with one another that were never intended to, which is really exciting. And just one last example is, what would this look like potentially if we had multiple drones in a shared environment? This is showing dynamic collision avoidance, but really for a high throughput screening challenge, we would have fixed stations that these drones would fly to, but this is showing conceptually rerouting. So what you're going to see here is the squares in the ground, and there's red, green, and blue, are where the drone is supposed to go. The circle is where the drone is actually currently located. And what you'll see is they intersect as the drones can dynamically avoid colliding with one another as the circle meets up at the square, if you want to show this video.
Don't be doing Rosbach.
And so what you're able to see from this is not only can one drone actually do a pick and place, you can have several drones working in tandem and actually avoid collision with one another or technically people. And remember, this is any autonomous vehicle. It could be a land-based instrument is actually more suitable towards lab environment as opposed to a drone, but it's really possible. And so really what we wanted to show in this presentation is as you're expanding your automation and you're looking from where you currently are to where you want to be, is if you look at devices from the component level and then how that component impacts the device, and then how that device impacts the overall system, and then how that distributed systems can actually work together, and then the possibility of integrating these distributed systems is there you can actually have the potential for autonomous assays. And so if you imagine from this perspective, if we have our high throughput screening system as one distributed system with all its peripheral devices, and then we have our select T, for example, that's generating cells, is if it was capable of sending an event, which it is when a plate is available, and we could integrate that with an overall controlling architecture, is that could actually initiate a new assay on a completely independent system if we have some plate transportation mechanism. And that's really geared towards what we're shooting for eventually is this concept of autonomous. So if you look at the trajectory of automation over the years, it was a long time ago now that things were computerized. And then it went through the step of where they're actually automated. And we're looking for now for them to both be sustainable, distributed, but really the goal is once again autonomous. And we're not there yet, and there's plenty of work to do. But we're showing through some of the work we're doing at NCATS and with many of our collaborators in both partnerships and SBIR contracts is we're really on the track for some really exciting times. And we haven't even talked about the actual data analysis piece, which is a huge part of this, but a presentation all to itself. So with that, I want to thank everyone for their time, and especially this is a complete team effort at NCATS, and none of this could happen without all of these components working together. Thank you very much.
Thank you, Sam, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on the screen and click the Send button. We'll answer as many of your questions as we have time for. So let's get started. First question is, what are key considerations to consider when designing a high throughput screening process?
Very first consideration, and this one really before you do anything else, is understand what you're even trying to automate. And what I mean by that is typically when these systems go off the rails is people try to have them do too much and they end up designing towards the very low use cases and introduce complexity. Is what we try to do is design something in a modular fashion towards the most common approach you want to take. So really a key consideration is understand your process that you're trying to automate. Don't make a system try to do too much, or especially don't design to the exception. But then also make sure you have modularity. And a key consideration is make sure you have complete control of the software, both from a perspective of being able to monitor what it's doing, but also from a perspective of actually being able to schedule it and be able to control it.
Thank you. What should someone look for when considering high throughput screening scheduling software?
In line with the previous questions, you should really consider the capability, once again, to be able to monitor and log every event, because it's hard to say when you know an event will become important and you might have an experimental design down the road that you might want to track something. And if you don't have the capability up front, it could really limit you. So one of the first step is make sure it has monitoring and monitoring control capabilities. But even furthermore, make sure it has the ability to have programmatic control, meaning you can get the scheduler to do something programmatically from an external application. Many of the schedulers available, both commercial and open source these days, have that capability, be it direct or even you can do plenty of things to game scheduling software. But a real consideration, especially when I shoot towards more advanced automation and more tricky experiments, is having a very fine level of programmatic control of the scheduling software.
Great. What direction do you see going forward for high throughput screening systems?
I think what we're seeing now is the days of the monolithic larger system, even the ones that we have back to the early 2000s, they're still in use and at certain places, but I think they're going away more towards a more modular approach where you can actually split up your entire process, that maybe it takes place on several systems, not just one. So I think it's a mixture of having this modular approach and also this nimble approach so it can be reconfigured. You can still, the libraries are still large in size, but in many cases more focused specific libraries, but there's more complexity coming with assays. And so you want to make sure that if you do design a system, you can keep up with that actual additional complexity. And that's something that we're seeing at NCATS, and also at some of our other folks involved in this space are sharing some of that those thoughts as well.
Okay, we have time for one more question. How practical is it really to consider having a drone in a laboratory environment?
Not at all. Not at all in its current state. But what this is shooting towards is this concept, again, of autonomous. And autonomous vehicles are more and more prevalent. They're already heavily in use now, and especially in inventory and tracking applications. And this concept of being able to actually do a pick and place, instead of just a monitoring type event or taking an inventory, is really transformational. So as it stands right now, it's not too practical, but there is a lot of work going on autonomous vehicles, not just drones. We consider a land-based vehicle. There's actually some other groups actively working in this space. In a very controlled fashion, if you can control the drone's flight path for a very specific use, it could have some practicality, but it's really about pushing the technical boundaries to get towards that goal of autonomous beyond just automation.
Well, thank you. I would like to once again thank Sam Michael for his presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through August 31st, 2018. You'll receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us. We hope to see you again soon. Goodbye.